Okay, great. So now that the recording message has gone, I'm allowed to speak. So thank you all for coming to this seminar. So really excited today because one of the main goals of the Catalyst series is to try and integrate the kind of amazing prowess that UCL has in basic kind of discovery research using animals, and also how these translate into the brilliant research that's also done in the clinic. And the two guests we have today are really, I think, at the forefront of this at UCL, and it's really exciting to have them both here today. So the first speaker is going to be Dr. Katharina Schmack, and Dr. Schmack's research focuses on psychosis, and her lab investigates the neural circuits and immune processes giving rise to hallucinations and other psychotic symptoms. The really exciting thing for us is she actually uses a cross-species approach and studies both patients and mice using kind of common behavioral tests. And our second speaker is Professor Tara Keck. And she, in her research, Dr. Keck facilitates and investigates mechanisms of neuroplasticity and their consequences for behavior during healthy aging or after extremely stressful events, as well as how neuroplasticity is disrupted during age-related diseases. And what's really interesting as well is that Tara has also now taken a foot into the kind of more kind of human and patient-y type focused cohort, and she's going to tell us a lot about that specific transition today. And so basically, I think we're going to have both of these talks. Both are going to be roughly about 20 minutes each. And so please feel free to ask questions in the chat or save up your questions for the end. But the way we usually do this is that we save the kind of discussion and questions until the end of both talks, just to facilitate discussion about both things. But if there's a burning question you want to ask after a particular talk, please just go ahead and do that as well. With that kind of brief introduction, I'm really excited to start off with Katharina, who's going to talk us about computational psychiatry across species to study the biology of hallucinations. So take it away, Katharina. Oh, I was too quick.
Thank you.
Oh, there we go. Okay, good.
Sorry, yeah, I didn't find the unmute button. Yeah, thanks a lot for this introduction, and thanks for the invitation. As Andrew already mentioned, I am interested in psychosis, and the reason I'm interested in psychosis is that psychosis is characterized by these very fascinating experiences, I think, which basically are perceptions of something non-existent. So in the case of hallucinations, people often perceive things that others cannot perceive, especially hear things that others cannot hear. In the case of delusions, people typically perceive, for example, threats that others cannot perceive. And to me, this really illustrates one fundamental aspect of how our perception works. And therefore, to me, understanding psychosis can provide insights into the basic mechanisms of perception. But of course, as most of you probably know, psychosis is not only interesting intellectually, but it's also a highly relevant condition, because it is the hallmark of many disorders, psychotic disorders such as schizophrenia. And these disorders are quite devastating in the sense that they often, they are frequent, and they start often.
Can you speak any faster?
Sorry? You want me to speak faster?
No, I think it's I think you're at a good speed, and I would appreciate if people don't interrupt the speaker. Thank you.
Okay. There will be also a recording that can be watched at different speeds, if that helps. Yeah, so what makes it quite relevant is basically that it starts early in life, and more often than not, it does not fully resolve. And what is particularly frustrating to me is that this has not dramatically changed over the past decades. The treatments we have somewhat stagnated, and especially the medications we have were all discovered by serendipity a long time ago, basically. And we still do not fully understand how and why the neural circuitry in the brain gets affected to produce the symptoms of psychosis. And for me personally, this frustration drove me from being a pure psychiatrist really into neuroscience, because in neuroscience, there has been a lot of progress. And we now have really amazing tools at our hands, and I will showcase some of these tools, that allow us to study the neural circuitry and behavior in amazing detail. The only issue is that to make use of these tools, we need to find ways of studying psychosis in mice, because that is where we have most of these tools available. And so this is kind of the overarching goal of our research, to find ways to study psychosis in mice and use that to dissect mechanisms that can then guide future development of treatment. And so there are two basic challenges in studying psychosis in mice. And the first challenge is that it's quite difficult to measure psychosis in basically any individual, but in particular also in mice. And that is because psychosis is characterized by these subjective symptoms, and we don't have any hard objective markers. And so I will today present you a framework how we started to address this and overcome this using what we call hallucination-like perception. And then in the end, I will briefly talk about another challenge that we have started to take on in our lab right now. And this is the challenge of, even if we can measure psychosis in mice in a meaningful way, we still need a way to then actually get mice into a state that recapitulates what psychosis in a good way. And I will briefly tell you in the end how we are making use of insights into the immune dysfunction underlying psychosis to actually get mice into a state of that can inform us about psychosis. So how do we measure something that is relevant for hallucinations in mice? And to do this, we started off with the basic definition of hallucinations. And if we look at what a hallucination is, it is basically a false percept that is basically in the absence of a causative stimulus, but that percept is not only false, but it's also perceived with high confidence or certainty. And we thought that this is something we can model in mice. And to do this, we came up with this task where basically mice, can you see my laser pointer?
Yep.
Well, thanks.
Yes, we can.
So mice are running around in a box, and there are three little holes in the wall. And so whenever the mouse pokes into the center hole of these three holes, we play a brief tone. And that means to the mouse that it has to go to the right. If it does so, it gets a water reward, a little drop of water. If it goes to the left, it doesn't get any reward. So this is half of the trials. Randomly interspersed is another type of trials, which are the no signal trials. And in these trials, the mouse pokes into the center, but now we do not play any tone. And now the mouse has to go to the left to get a water reward. If it goes to the right, it doesn't get a reward. And this gives us the perceptual choice of the mouse. Now, we also wanted to measure confidence. And to do that, we used a trick. And that trick is that we basically did not give them the reward right away, but we delayed it for an unpredictable interval of time. And also sometimes when they were correct, we would just not give it to them. And so the effect of that is that the mouse never knows whether and when a reward will arrive. And as a consequence, it has to make an active decision how much time it is willing to invest. All right, so in this framework, we can define hallucination-like perceptions as trials where we don't play. So basically, what we can measure with this task is hallucination-like perceptions, which we can define as false alarms. That means trials where we do not play a tone, but the mouse nevertheless reports hearing the tone. And it also does so with high confidence, as indicated by a long time investment. Okay, so the first question we had to tackle is like, are mice actually able to do this task? And the good answer is yes, they are. So when we look at their choices, so this is a psychometric curve here. So on the x-axis is the signal intensity, so how loud the tone was over background noise. And on the y-axis is the percentage of trials the mice reported hearing a signal. And what we can see here is that whenever the tone was very loud, mice actually almost always correctly reported hearing that tone. As the tone volume decreased, this percentage decreased. And importantly, even if we did not play a tone at all, the mice still reported hearing tones on a significant proportion of trials, approximately 15% of trials. And this is good news for us because this means we can induce these false alarms, which are the interesting trials for us, because that's what the first kind of condition for our hallucination-like perception. All right, so that's their choice behavior, their perceptual behavior. Now, what about the confidence? And here we first, if we want to know whether their time investments do reflect confidence, we first need to have an idea what confidence should look like. And for this, we recur to this model of statistical decision confidence. And basically, this model simply says that confidence should reflect the probability of being correct. So if the probability is high, we should be very confident. If the probability is low, we should be not very confident. And because the probability is a construct that it's a mathematical construct that we can calculate, it's quite straightforward to do that. And when we did that, we can plot this probability of being correct. And this is what you're seeing here. And then we can overlay the actually observed time investments from the mice. And as you can appreciate, I hope, these look very similar to these predicted confidence values. And this is the case for one mouse, but we saw very similar things in all 8 mice, where basically we found that the confidence model predicted the behavior, the time investment behavior really well. Now, just because we call it hallucination-like perception, it doesn't mean it has to do anything with real hallucinations. So what we did next is we wanted to know if we push mice in this task with manipulations that we know are related to hallucinations in humans, can we actually push these hallucination-like perceptions in the right direction? And so the first thing we did, is based on the observation that human hallucinations are related to strong perceptual expectations. So we know that people with hallucinations, they rely more strongly on perceptual expectations. So what we did, we mimicked the same in mice, and we had a version of the task where we induced perceptual expectations, basically by just having different blocks where the amount of trials where a tone was played was different. And what we could see is that the more signals we played, or the higher the expectation was that there would be a signal, the more false alarms the mice had, and the more confident they were in these false alarms. And this is just showing the psychometric, showing that actually these expectations shifted the psychometric and led to this perceptual bias towards perceiving more signals that were not there. The next thing we did is ketamine, because it's known that ketamine can induce some psychosis-like experiences in people. So when we treated mice with ketamine or with vehicle, we saw that ketamine also induced these false alarms and increased false alarm confidence. And again, and this is truly remarkable, I think, this was explained by a shift in perception. So very interestingly, mice did not have more errors in general. They were still pretty good under ketamine here for these very loud tones. But as you can see here, they had a specific effect on these more difficult trials, or on these no signal trials. So they had a specific effect on having more false alarms. But of course, to really know whether something is a good measure of hallucinations or not, we need to directly relate it to hallucinations And because we cannot do that in mice, because we cannot ask them whether they have hallucinations, we went and translated this task back into humans. Very similar task, we play tones and ask people whether they hear the tones, and then ask them how confident they are. Here we can ask them directly how confident they are. And what we also did, we also measured their tendency towards hallucinations. So this is a general population recruited on Amazon Enter. But as some of you might know, there's actually quite a spectrum of hallucinations, even in the general population. And this is what we observed when we used a questionnaire to measure that most people did not have a lot of hallucinations, but there was quite a wide spread of this. And there were a few that really had a lot of hallucinations, even in their daily life. And what we then found was that these self-reported hallucinations were positively correlated with the false alarm rate here in the task, and with the false alarm confidence in this task. And what was really nice was that this was somewhat specific to hallucinations. So when we looked at other measures of psychopathology, so depression or anxiety, we found that hallucinations alone explained the behavior in this task best. So what I have shown you so far is basically we developed this task to measure hallucination-like perception. And I showed you that mice can do this task. And I showed you that in mice, hallucination-like perception increases after hallucinogenic manipulation. And in humans, hallucination-like perceptions correlates with self-reported hallucinations. So what this suggested to us is that maybe our task is tapping into some mechanisms, or at least into some engaging some processes that might be relevant for hallucinations. And now looking at the brain in this task might inform our understanding of the biology of hallucinations. And so to look at the brain, we started with dopamine. And the reason we started with dopamine is because the reason is that we know that increased dopamine in the striatum is related to psychosis. We know this on the one hand because all the antipsychotic drugs we have are basically working antagonists of the dopamine D2 receptor. But also we know from imaging studies that people with psychosis and even in early stages of psychosis have an increased dopamine turnover in their striatum. But what we do not know is why too much dopamine should lead to these very perceptual experiences in psychosis, such as hallucinations. And now we have a really good framework to test this, and that's what we did. But before we went into the brain, we first wanted to have a really good theoretically grounded hypothesis of what to expect and what to look for. So from a theoretical point of view, Bayesian perception implies that when there's an imbalance between expectations or priors and evidence or the likelihood, then hallucinations emerge. And we simulated that using the simple model. And we found that this model really nicely recapitulated our task. So here at the left is, there's a error here, but at the left is the model. And I hope that you believe me that the psychometric looks very similar to the mice, and also the confidence that the model produced was very similar to the observed confidence in the mice. And we validated this along other dimensions, and the model could reproduce the behavior of the mice quite well along several different domains. So this model makes an interesting prediction, basically two different predictions. The first one is that if we think about how dopamine could relate to hallucination-like perception, one possibility this model predicts is that maybe dopamine encodes expectations about perception, or about whether a signal will come up or not. And in that case, too much dopamine would shift the amount of all signal choices, and would increase all these signal choices, and thereby lead to more false alarms and also more confidence in these false alarms. Alternatively, dopamine might encode expectations about reward. And in that case, the model predicts that this would lead to more errors in general. And this would be the mechanism by which, or the algorithm by which false alarms and confidence in false alarms increases. Okay, so with these theoretical predictions, we then went and measured dopamine. And to do this, we used these very nice sensors. So it's a really nice tool that was recently developed. So basically what that is, it's a protein that we can express in the brain using a virus. And this protein has this nice property that whenever dopamine binds to it, it emits fluorescence. And we can then implant a glass fiber on top of the protein, or in the area that we're interested in, and can measure this fluorescence as an ongoing measure of dopamine release. And this is what we did in two regions, in the ventral striatum, because this is a region that is known to be important for reward, and in the tail of the striatum, because that is a region that is important, or there's some evidence that this might be a region that is important for perception. And this was obviously guided by our hypotheses from the model. And what we found was really exciting, I think, because what we found is that when we now what you're looking at are these dopamine levels over time. So on the x-axis is time, and on the y-axis is the signal strength of the dopamine. And what you can see is here 0 is the time point where we play the signal. And what you can see is that even before we play a signal, dopamine was higher when the mouse would go on and have a false alarm, as opposed to trials when the mouse would correctly not hear a tone. And this was the case in both of the regions that we looked at. And this is just to show this was very consistent across mice. So this is the data for all the mice. And what was really nice when we further looked in these false alarm trials and compared the ones with high confidence, or the hallucination-like perceptions, with the low confidence false alarm trials, or the mere errors, we found that dopamine was particularly high on these high confidence false alarm trials. And this was really a linear relationship, as you can see here. So on the x-axis is now the dopamine, and on the y-axis is the probability that the mouse will report a high confidence false alarm on this ongoing trial. And what you can see is that, yeah, this is linearly increasing. So the higher the dopamine is at the beginning of the trial, the more likely the mouse will have this hallucination-like perception. And this was, again, like very similar in both of these regions. But then when we looked more into the data guided by our modeling, we found that there were different explanations for this. And this is a very busy slide, but I just want you to focus on the big picture here. What I'm showing you here at the right, at the left, is basically the dopamine signals in the ventral striatum, and at the bottom is the predictions from our model for these reward expectations. And I hope that you can appreciate that they look very similar. So basically, dopamine in the ventral striatum, in this reward region, looks very similar to reward expectations. Whereas in the tail of the striatum, this perception region, we found that dopamine looked very similar to these more perceptual signal expectations from the model. Suggesting that although in both of these regions, dopamine seemed to kind of precede or increased dopamine preceded these hallucination-like perceptions, in the ventral striatum, this might have been explained by an encoding of reward expectations, whereas in the tail of the striatum, this might have been driven by more perceptual expectations. So, but then we wanted to know, is dopamine actually really causal for these? Can we drive these hallucination-like perceptions by manipulating dopamine? And so what we did, we used optogenetics, which is another amazing tool of systems neuroscience, where we use this other protein channel rhodopsin, which is a channel that opens up whenever light is, when we shine a light on it. And so you can see here that when we use mice that have this channel rhodopsin specifically in neurons that release dopamine, you can see that if you shine a light on them, they release dopamine. And so this is what we did in the tail of the striatum. And now what we found was that whenever we optogenetically elevated dopamine, mice had more false alarms, and they were more confident in these false alarms. And remarkably, this was rescued by haloperidol. So here you can see after vehicle, mice, the optogenetic elevation of dopamine leads to more false alarms and more confidence here. When we now pre-treated them with haloperidol, you can see that this optogenetic dopamine elevation had no longer an effect in both of these conditions here. So just to summarize what I have told you, I showed you that expectations do link dopamine and hallucination-like perception from an algorithmic perspective. I then showed you that elevated dopamine in the striatum precedes these hallucination-like perceptions. That's what I call a correlational link. And then finally, I showed you that if we increase dopamine in the tail of the striatum, we can induce these hallucination-like perceptions, and this can be rescued by an antipsychotic drug. So how does this all make sense together? And this is the current model that we are having about this. So the idea is that basically dopamine might encode expectations and thereby explain why too much dopamine might lead to hallucinations. And the way we're thinking about this right now is that we know that the striatum is the site where inputs from the cortex and inputs from the thalamus are integrated. And dopamine basically is a neuromodulator that modulates the integration of these two. And we think that maybe the thalamus conveys the more sensory information about the stimulus, whereas auditory cortex is concerned with these more with expectations. And now these two are integrated, and when there's too much dopamine, then for some reason this balance is shifted towards more these auditory cortex inputs and towards relying more, and this leads to perceptual experiences that are relying more on expectations, and that explains why hallucinations emerge. But I think the other really exciting kind of take-home message from this work is that I think we now have a framework that allows us to actually study hallucinations across species. And I think that the general assumption here of this framework is that if we believe that neural circuits, impairments in neural circuits underlie symptoms in psychiatry, as shown here, and if we also assume that neural circuits instantiate some computations that link these circuits with experience here, but also with behavior, then we can actually think about how to measure them. And I think the exciting thing is here that we can think about tasks that capture these computations, similar to what we did here with our hallucination-like perception task. And we can develop these tasks both for humans, where we can then link them to these psychiatric symptoms. And we can also develop these tasks for mice, where we can basically then go back and gain insights about the neurocircuitry that is driving or underlying these psychiatric symptoms. So that's kind of the bigger picture, and we're currently following up on this in our lab, where we are looking at neural circuits that might inform the development of new treatments. And basically, you might wonder, why did we study dopamine in the first place? Because I told you that we already have all these drugs that do target dopamine. So what's the point of studying this? And I think the thing is that we do have drugs that target dopamine, but they don't do this in a good way. And here's why. So we know that too much dopamine in the synaptic cleft leads to psychosis, and then these, or that's our model at least, and then these antipsychotic drugs to block this. And what we know is happening is that this over time leads to an sensitization. So basically we think that this leads to a state where dopamine receptors are expressed more and in a more sensitive form. And now we have two problems. We have too much dopamine in the synaptic cleft, but we also have an oversensitive dopamine receptor system. And so the way around this, this obviously results in some of these things we see in the clinic, which is that people who go off antipsychotics get these rebound psychoses that might be even worse than their initial psychosis. And also over time, many people need more and different antipsychotics to actually counteract these sensitization effects. So the solution to that is to actually modulate the dopamine release, because then we don't get the sensitization. And that is why we are now using this framework to look at upstream mechanisms of dopamine, basically a dopamine modulating neuron populations, using our task and measuring these neuron populations. And what we hope that what we can do then is to use molecular tools to really identify potential treatment targets in these populations. And I think I just wanted to briefly mention, I'm not sure how I'm with time. So please interrupt me if I'm going over time. I'm a little confused with a break in between, but I'll just go on until I get interrupted. So I'm almost done. So the other thing that we're doing is concerned with this, with my frustration of not being able to have a good causal model of psychosis. So we're really struggling to induce psychosis in mice in a good way. And the reason for that is actually not that we don't know that we don't know anything about the causes. It's actually that we know too much about the causes of psychosis or psychotic disorders. So we have identified genetic variants, we have identified a few environmental factors that all increase the risk for psychosis. The challenge is that all of these factors increase the risk by just a tiny amount. And even if we find something that has a larger effect, usually it's happening really far apart in time from the actual experience of psychosis. So it's really hard to get to the biology that then happens in psychosis using these causes. And so the solution for this is obviously to find the common endpoint where all of these factors or some of these factors converge. And I think the ultimate endpoint is the brain, but there's a lot of evidence, in my view, that suggests that before the brain, the immune system might play an important role. And basically, there is this hypothesis that an overactive immune system might start to target the brain and specific circuits in the brain, and that might then lead to psychosis. And so what we're currently trying to do is we work in people with psychosis, where we try to identify some of these immune alterations, and then translate these into mice, where we try to generate a state where we immunize mice, or we create a state where we immunize mice against these brain circuits, and then see whether their behavior changes. And this will allow us to actually tap into these immunological mechanisms of psychosis, and to generate this model of psychosis, this etiological model. And so just to have very high level summary here, I think what I showed you, that we have these two challenges. The first one is measuring psychosis in rodents. And I think we have started to provide some evidence that we can use these frameworks and do something like hallucination-like perception to actually be able to get to the circuits that are relevant for psychosis. And the second one is this induction of the state of psychosis. And I think we are currently exploring the idea that we can use brain-directed immunity to translate between humans and mice. And so what I hope to see in 2052 is basically that with these insights that we and the field will generate over the next 30 years, that we will get to actual mechanistic treatments. So a mechanistic understanding that then will lead to new mechanistic treatments, such as circuit-based treatments, for example, treatments that target dopamine populations upstream, populations of upstream of dopamine, or that we get immunomodulatory treatments that are directly targeting very specific processes that might be involved in psychosis. And with that, I would like to thank all of you for your attention. I also would like to thank my the Keppich lab and Adam Keppich in particular, who in whose lab I did this the work on hallucination-like perception. This is our psychosis collective right now. And I look forward to any specific questions right now and then to the discussion in the end.
Brilliant. Thank you very much, Katharina. That was excellent talk. I think
you.
maybe in the interest of time, unless we have burning questions about the talk, is it okay if we leave, we kind of try and get them all together at the end?
For me, it's great.
Great. I've never been called a boomer before, so that's it's a first. It's nice. From that guy. Anyway, cool. So next up is Tara Keck. I think we're going to take a slight shift from the kind of focus on animals going directly to translation, and now we're going to talk about the actual translational side of this a little bit more. So thanks, Tara, and I'm very excited about your talk.
Yes, thank you, Andrew, and thank you again for the invitation. As Andrew said in the introduction, a lot of the work that goes on in my lab is related to aging and older people. So I decided today that I would take sort of a detour and talk about a project that I've been working on that's been a bit of a side project for me that's been continuing to grow. And this is with young people, specifically university-age students. And this is in the early stages. As you'll see, we've done a few iterations of this, but we're still collecting data. And so we're also really enthusiastic for any suggestions or ideas related to the project. And what I'm going to be talking about today is very much thinking about mental health and how mental health can be implemented at the level of communities. And I'm going to talk a bit in a minute about why we think that is really important. So when we're thinking about mental health, we know that if you pick up any newspaper, if you talk to any student, that mental health issues for young people, in particular university students, has increased. And what you can see here is that, so this is from the government, so there's been a 6-fold increase in student ill mental health since 2010. And so this is students that are reporting a mental health condition either on their applications or to their universities. And you can see that this goes up, but the numbers are still relatively low. But this is reflecting two things. One, that there's a higher incidence of students who are experiencing mental health conditions. And also that what is, I think, a very positive thing is that some of the stigma associated with actually reporting and declaring these conditions is starting to ebb away, where people are more likely to disclose them. And so you can see that this ends sometime around 2019, so right before COVID. And these numbers have dramatically changed since COVID. And so what we have here, sorry, my Zoom is in the way, there we go. So this is a slightly different measure, but basically if we start going from 2020 and beyond, 21% of students have a formal mental health diagnosis at universities. And when surveyed, 34% have some sort of psychological issue that they feel that they should get help for, but don't necessarily always do that. And so we've already had this issue of this increasing amount, and this continues to get worse. And obviously, when we think about the COVID-19 pandemic, this did not help anybody's mental health, but particularly young people. And so this is just one survey, which is asking university students how COVID has affected them, positively, negatively, or no change. And when you, and students, this is in 2020, we're having issues across the board. But in particular, when we looked at mental health, many students were saying that the pandemic and classes online and the interruption to their lives has been fairly negative for their mental health. And so this was sort of the starting point for this project. So we started thinking about this in summer 2020. when we recognized that students were struggling, and as we then saw students coming back and trying to do their year in 2020 to 2021, which ended up then being largely online in the spring term, we really wanted to put something in place to help address this. And so the question we asked is, what could we do to help our students adjust post-COVID, and particularly in relation to their mental health? And so what we decided to do is we looked into I based this off of some work that I had previously done, and thinking about COVID-19 like an extremely stressful event. And so basically it was something that was collective, it was something that affected a lot of people and didn't affect everyone in the exact same way. But that allowed us to look to the humanitarian sector. And so in the humanitarian sector, there's a large literature on mental health where, following things like earthquakes or floods or tsunamis or war, refugees, things along these lines, that people who are experiencing extremely stressful life events have mental health responses to this. And there's been a lot of work done by a number of intergovernmental agencies, universities, academics, looking at how it is that we can support people's mental health following these humanitarian events. And so we took this as sort of the basis for our project, and thinking about COVID-19 as a stressful event, and how do we help our students recover from this following. And so we looked at the IASC guidelines. Just to give you a brief overview, these are basically mental health and psychosocial support in emergency settings. And this is considered, for the past 15 years or so, the gold standard for humanitarian settings. The basic principle behind this is not providing high-level mental health care. I'll talk about that more in a second. But the principle is, doing no harm and making sure that people have basic levels of support. And a big part of all of this is trying to stop the focus on PTSD or post-traumatic stress disorder or trauma, and more recognizing what it is, the support that people tend to need. Because following extremely stressful events, a small number of people develop post-traumatic stress disorder or trauma or traumatic stress responses. Most people actually just need basic levels of compassionate care. And the idea of developing these more severe mental health disorders would be associated with previous exposure and other risk factors. So we were sort of trying to get at sort of like a basic care that would be effective for many of our students. And a really important part of ISC is stopping the harmful psychological debriefing. This is where, when you ask people to recount something that's happened to them, which at this point we recognize potentially causes a lot more harm than good. So, sort of as a second step here with thinking about ISC guidelines, we have the mental health and psychosocial support pyramid. And so what this reflects is that these are the different levels of support that someone needs following a stressful event. And so we were thinking about where in this pyramid we'd want to be targeting and be supporting our students. And so the bottom of this is basically providing people with basic services and security. And so this is making sure that people have their basic needs met. And when we first started looking at this in 2020 and 2021, at that point, safety was a concern, but did seem to be being managed. So we were less concerned about this now. As we continue to think about this type of mental health support for students, there are additional economic factors that are kicking in, where basic services, things like heat poverty or food insecurity, are starting to hit our students. So this is something that we're going to consider revisiting in the next iteration. Following basic services and security, strengthening community and family support. So community type networks are really critical for people, both in their recovery from a stressful event and in building resilience for them. And then we have the upper groups. So here we have focused non-specialized support. So this is basic mental health care, but by professionals. And so this would be, for example, our well-being advocates in our departments, people who have had some training but aren't necessarily clinical psychologists. And then the top is specialized services. And so the idea behind this is that if you're meeting the needs at the bottom, then fewer and fewer people will need these specialized services. And so what we're trying to do is look at places where we can start to provide service that reduce the number of students who need these more specialized care. And so as we think about this, the key resilience factors that we're concerned about is we know that people are less likely to develop more severe mental health conditions following stressful events if they are feeling safe, connected to others, and calm. So that's this basic security at the bottom. The second is that they have access to social and emotional community support. And this is where we saw that there was actually a nice gap in what was available at UCL, is that these community networks weren't being trained. And what I have written here is this 25-50-25. And so what we know that's sort of loosely true across a number of mental health conditions is that following an extremely stressful event, 25% of people sort of dependent on risk factors that they've previous experiences, other risk factors that they have, are going to need specialized care from someone like a clinical psychologist or a psychiatrist. 50% of people, if they have this social, emotional, and community support and have their basic needs met, will most likely recover well with time. They won't end up needing this specialized care if this community support is in place. And another quarter of people or so are actually just quite resilient and bounce back from things, even if they don't necessarily have the community support. And so who we're really targeting in our student group is these students in the middle who, as they experienced COVID, but then as they continue on through university and experience stressful events, making sure that they have community support in place so that they're able to recover and be resilient and be happier with time. Again, having a sense of belonging in their community and being tied to one another. And so this is something we were targeting as well. And finally, what we know is that people who do need this specialized care, so if you do end up going into some form of therapy or using different types of medications, people who have support in their community have better outcomes following this specialized care. So for us, it really did seem like targeting the community levels of support would be a great place, and also a place where we actually had some influence. So we weren't able to necessarily hire more mental health specialists into our degree programs or into our faculties, but this is someplace we could actually train people, and they can be trained in a small amount of time. So we're thinking about this at the community level because we think that there's a lot of potential for diffusing potential issues. And then we thought through, well, who do students talk to first in their communities? And we took some data from student surveys. We chatted with some students through some informal focus group type events. And as much as we like to think that they're coming to lecturers and tutors or wellness teams, when something's difficult, students are almost always first speaking with their friends. And as we were talking to students, this was who they would first disclose to. And then they immediately told us that that many of them are very nervous when someone discloses to them because they don't know what to say. And they don't want to, make it worse. So I think most people are coming to this from a very positive, wanting to help point of view, but they haven't had training and they're unsure what to do. And what we know is that these first interactions are important. So for somebody to disclose something that's distressing to them, they get into the issue of if it goes badly the first time, they might be nervous to disclose to somebody else. So it's both for providing support, but then also in the event that what we have here with students is that in addition to being able to provide this basic non-judgmental support, if they identify students, we can identify students who need to get into referral pathways and get to our wellness teams. And so what we did is we developed community level peer-to-peer training for first year students. So during this period, we train them in order to provide basic support to each other. And the idea is that we want to teach them to provide non-judgmental support, that because they'll have this community within their cohort, that hopefully in some instances will prevent the need for more specialized care. But as a part of this, and I'll go through the outline of the program in a second, it also helps us as faculty have the students identify people who might need more specialized care and get them into referral pathways. And then finally, also it helps support people who are receiving more specialized care, which is a fair number of our students who are seeing mental health specialists and making their outcomes also better. So in addition to developing this program, we wanted to add one other thing. And so this is, as Andrew said, I study neuroplasticity and think about memory, especially following stressful events. And so we wanted to think about the neuroscience of this and how this might be incorporated. And so we know that having a basic understanding of how a traumatic memory is formed and then also diminishes with time, can be helpful for both the person giving mental health support and the person receiving it. And I'll talk about that more in a second. And so we, in this training, included a few sessions on traumatic memory, although we just call it stressful events, and why particular recommendations for recovering from these things help with your brain rewiring, and so how it is that that the students can recover. And we also emphasize why recovery can take time. And this is really important because many of our students, and in mental health in general, will start doing things, and when they aren't getting better within a few days or even a week, they can sort of become upset by that. And so understanding a bit of the brain wiring allows them to see, oh yes, it's going to take a little bit longer than, you know, a day or two. So the overview of our training is that it's interactive. It's during induction week for our first year students. And the first iteration with it was more lecture-y, and it gets more interactive over time because that seems to be working better. And what we're in essence teaching people is psychological first aid. And so this was developed together between the United Nations and WHO for humanitarian settings. And all it is basically providing basic support for people in distress with a focus on non-judgmental communications, listening skills, and providing support and linking people with the help that they need. In addition to psychological first aid, so basically good listening, we also are introducing the neuroscience of memory and why certain approaches would work. And we then also have a section which was introduced a bit later, which has been really effective with our students, discussing the difference between emotions, which are healthy, and mental health conditions, which are something to maybe to keep an eye on. And so through that, we give a brief overview of when students need to make sure that their course mates or their friends get help. So basic signs of depression, extreme stress, post-traumatic stress disorder, and then also suicide and self-harm. So we have these open conversations about, like, this is when it's time to go and seek help from a wellness tutor or a mental health specialist, or in some instances, the hospital. And so I'm just going to take you through a couple of the slides and then some of the first preliminary outcomes that we have. So this is an example of how it is that we are interactive. So we ask our students to start thinking about if you had a stressful event happen. So if you got a low score on the first quiz you took, what would you need as the person to have this happen to you? And students give us a range of answers. And if you think about this yourself now, different people like to do different things when bad things happen. And so what we do is we ask them to say this out loud, and we always get a wide range of answers. So some people want to forget about it and go out with their friends. Some people want to go over the quiz and come up with, you know, a study plan for next time. Some people want to go do something like exercise. Some of our students say that they want to scream or cry. Some people call their parents. Some people talk to a friend. There's a wide range of things that our students say that they want to do. And so we use this as the basis to say, well, if someone said this to you, that this happened, what would your first question be? And so what we really work with them on is asking a question that, given all of the issues or all of the ways that people wanted to respond, what is a question that you could ask that isn't sort of pushing your idea of what's right on someone else? So if you had failed a quiz and you wanted to go through it point by point and figure it out with someone, but your friend wanted to go and forget about it and never think about it until, you know, coming back to it several days later, suggesting this might not be so helpful. And so similarly, this also gives us the opportunity to talk about what can be very well-meaning but come off as judgmental questions. So what we always say is, someone says, you know, well, I failed a quiz, and we ask them, well, you know, what if I respond, well, I can help you study next time? And we talk about, you know, the judgments and assumptions in that statement, which is very well-meaning in many instances, but can have a very negative effect in that you're assuming the person didn't study, you're assuming that you know more than they do, all of these things that make someone feel judged and hurt, when really what they need is open-ended mental health care. And then just another example in terms of thinking about stressful memories. When we're talking about the neuroscience of this, we don't get into this to the nuts and bolts. We aren't talking about which receptors are activated or which neurotransmitters are released. But we give these examples of real life things, you know, encountering a snake that's quite that's a scary situation, and, you know, what happens. So basically, you know, a negative emotional memories are strong, but then over time, these memories can be either reinforced. So if you were to come back to this area the next day and you see a snake again, you're going to be very afraid of this tree or the area by this tree. But if you come back over and over, slowly over time, your memories will be forgotten and become extinct. And so, you know, this first iteration is really important. And so this is the type of level of detail we're giving students. These, again, are first-year students and not necessarily not that far along in their biology yet. And then finally, what we talk them through is, you know, basically stress management, but basically we're continuing with daily routines, try and exercise, maintain a regular sleep cycle, avoid drug and alcohol use as a coping mechanism, and considering limiting screens or socials or other forms of stress. And so we go through these things and one by one talk about how each of these are linked to memory formation and how each of them are actually helping you change this traumatic memory from something that's very stressful and emotional to something that is no longer emotionally charged. And so this is sort of the overarching view of this. Through these, we've been then doing some basic surveys with the students. And so we've asked them a number of things. And so we have some students who've taken our neuroscience one and some students who have taken it before there was the neuroscience aspect to it. And this is the confidence that they have following the training. And in blue, it's without neuroscience explanations, and memory explanations in red is with. And so we asked them, you know, how confident do they feel in the ability to support other people? And you can see that they, when we do the baselines are somewhere around 2. So these go up in both instances, but more so when they understand neuroscience aspects. Similarly, in knowing what to say and do to help, we see also a stronger effect, but an increase in both groups. The ability to listen in a supportive way, they do feel like they're able to do that. Knowing what not to say to cause harm. And I think that for the students, this is very important to them. A lot of them who really do care about their friends really are worried about causing harm. Knowing how the brain recovers from stressful events, obviously, they're doing much better on that with confidence after learning it. And the ability to reassure people that things will improve with time. And so we are seeing that this is effective with our students, but this is actually just looking at it immediately after. And as I said, this is still in an early stage with our proposal, or sorry, with our program, and we're now trying to track mental health outcomes over time. So as a sort of overview for our outlook, the preliminary results, so we've been working on something similar with collaborators in the development sector, that when people are using this neuroscience-led mental health training with clinical psychologists and first responders, we actually see that there's a compliance for the people who are receiving it when they have these neuroscience explanations. So when they recognize that going for a walk or trying to get their sleep in order, what that's doing to their memory, they are at least in first instances reporting higher levels of complying with what's been recommended to them. If we come back to our student feedback is extremely positive. This is self-reported and obviously to some degrees biased because we're asking, but the students do feel, many of them have commented on the program and that they feel like it's because it's an induction week that it does seem like we're taking their mental health seriously, which is very important to them as on the whole. And so now what we're trying to do is we're trying to track outcomes related to, you know, what are the students doing, how are they talking to each other, and their overall well-being. Obviously, in the first few cohorts here, it's a little bit tough because they have not had who to compare them to is a bit tricky. But we are trying to come up with some measures for this. And we're doing this for students who have received the training versus their classmates. So to date, we've just been doing this with neuroscience students, and we're trying to compare them with biomedical students, but we hope to expand this beyond neuroscience students in the near future. And I'll just thank, so all of this was developed in collaboration with my collaborator at the University of Tokyo, Takashi Izutsu, and with the support of people in neuroscience, Martina and Paula, who are critical in our teaching, and also Subtiara, who is one of our wellness staff in Faculty of Life Sciences. Thank you.
Thank you very much, Tara. That was fascinating stuff. I want to open up for questions, but as people are thinking of their questions, I thought the main thing that struck me that was, I think we've almost got the perfect mix here, because we've got a kind of frustrated clinician coming to the basics to try and work out the core faults to help create treatments, and then we've got a kind of basic neuroscientist using the tools to try and actually make real change in the community, which I think is a really nice example of how you can go in both directions using the expertise you've got in either case. Are there any questions from the audience? I have also obviously got a lot of questions, but I don't want to hog the limelight. Have we got any here? No, maybe. Okay, if you have a burning, oh, yep, Essie.
I had a sort of a question that might be slightly left field to Katharina. So we know that there are big differences, individual differences in how much different individuals have insight about their psychosis and how sort of accepting they are that they are actually having psychotic experiences as opposed to something else. And we know that this also relates to how successfully their psychotic experiences can usually be treated and managed. And I'm wondering whether you have any sort of theories as to whether that relates to the extent to which they may have these deficits that you reported or differences that you reported, how pervasive they are, or do you think it relates to some other neurocognitive phenomena altogether, which is sort of outside the sort of phenomena that you are investigating?
Thanks. No, it's a great question. I think there are two kind of aspects to that. So I think the first thing, obviously our model is like a very simplistic model, and it obviously is oversimplifying things and only capturing this particular perceptual aspect of the story. So I think one aspect that our model has not captured that's probably very important for insight is kind of trust and more of these social domains of how much do you actually believe what others are telling you, and especially how much do you believe what doctors are telling you. And I think that relates a lot to your experiences, and it's probably the wrong model to look for that at the neurobiological level. I think that this needs a kind of more holistic societal perspective or psychological perspective. But I think the aspect that our model can capture, and that might be related to insight, and we haven't looked at that, but it might be an interesting avenue to embark on, is basically, so I think this confidence measure that we're measuring, and obviously this varies across trials, and this might vary across people, and also in general, how much confidence they put in their perception might vary. And of course, it's one hypothesis would be that people who experience these things with extremely high confidence, meaning that their perceptual experience seem extremely real to them, like not real in the sense that they are indistinguishable from other experiences. I would expect that this obviously impairs in what we call insight. I mean, also, I don't like the term insight so much because it seems like, I think from the subjective perspective, and that's the entire point of these models of hallucination like perception, is that the perception is like real. And I think this confidence part can exactly kind of put a number on this, how real it feels to people, and that might be correlated with what we call insight in the clinic. So yeah, I think it's a great question. Thank you.
Great, thanks.
The one thing I was thinking about in terms of this, just listening to both of your talks, and especially Tara's at the end, I just maybe a question for both of you is that, like, I guess Tara's stuff is showing really, like, at face value how difficult it is to kind of put this detailed kind of very mechanistic knowledge we have into a setting. And when you're creating these programs that are educating kids on how to interact with people, we're using extremely basic neuroscience knowledge, and even that basic knowledge is enormously successful. And so I'm wondering, like, to what extent, like, where is the correct level currently, right? Because we've got Tara on one end using like stuff we already know, and we've got Katharina at the other end being like, in 2052, what we're going to know is going to be even better, right? And so where do you guys have an opinion on that? Like, where do we sit, and what should we be focusing on in those kind of ideas?
Tara, would you like to start?
I can start, I guess. I mean, it's a good question. It depends very much on the question, right? I mean, I think that when you're looking at a drug target, well, that's something that we need very specific mechanistic, information for. But I think that when we're starting to think about things like mental health, but I mean, like, mental well-being is maybe the right word for this, or ways to start interacting with people, particularly at a young age, to talk about these things we definitely have, you know, I wouldn't say we could always use some more information, but we have enough information to talk about this sensibly and to start people on, you know, reasonable paths. I think that one example of this is, you know, is it useful to know the mechanisms of everything? Maybe, like, but do we need to know all the mechanisms, exactly the effects social media is having on brains, to say like, huh, maybe this isn't the best idea? Like, I don't know that we need to know all the detailed mechanisms in that instance to say, like, you know, to sort of use first principles and make good guesses. And in those instances, we're not going to have that data for a long time. So it's important that we start to make guesses now. When we start thinking about targeting something like schizophrenia or other forms of very specific drugs, well, we really do want to know the information and not just be saying, yeah, well, we have to solve it, so let's just put some chemicals into people and hope for the best. So I think that there is a balance of, you know, what damage are you doing, how obvious is the problem, and how long is it until we get good data.
Yeah, it's also interesting to see like even how little direct neuroscience input is not present, and when it is input, it can make a huge difference. And I found that incredibly interesting. Yeah, Katharina, do you want to?
Yeah, I mean, I think both is important. Like, it's basically in all fields, like we need mechanistic understanding, and at the same time we need to put that mechanistic understanding into practice. And I think there is probably, I mean, both of these things are lagging behind. I think there hasn't been enough, as Tara was showing, like there hasn't been enough work done, or that basically just takes very basic insights and tries to get them into the community, into people. And I think this is true for many aspects. I mean, the schizophrenia treatment is another example of that, where we have drugs that are effective based on clinical trials and everything, but still the outcomes are not what we would like them to be. And there's a big issue about like how to deliver treatment, how to make sure people stay engaged. And I think this is really important work that needs to be done. On the other hand, I think we need mechanistic understanding. I mean, I think it's also just like a human impulse. We just want to understand what's going on. And I think this was also important, an important insight, that basically, if you have any kind of intervention or treatment, it will just work much better if you can explain what is going on and why it is working. And I think the other aspect is, so it's a psychological effect, basically. So you need to understand why it's working to make it work even better. And the other thing is, I think this mechanistic understanding also allows us to then make gradual improvements, because I think if we have a black box treatment, might be super successful, but if it doesn't work in some instances, we have no idea how to improve on it, or where to actually, where to even look for things. Whereas when we start to have some kind of mechanistic understanding, and we see in some circumstances it's working, and others it isn't, we have a good guess like how to make it work even in these circumstances. So I think these are the two aspects, and I think they both go very closely in hand. And I think we just need people on doing like all of these things, depending on what they are most passionate about.
That's two really great answers. Thanks, guys. I guess we're kind of running out of time. I'm also knowing that we have people who can't stay until the exact 15:15. Maybe one thing to just kind of end on, if that's okay, would be the whole goal of this, right, is to increase interaction between human and animal researchers. And I'm just wondering, if there was like a kind of golden thing, what was the one thing you wish that was here at UCL currently that would aid this kind of more interaction, or help you personally through more interaction with the kind of interdisciplinary side of UCL? That's a horrible question, but I'm going to throw it at you anyway. Oh, it's too horrible a question.
Well, I think that some of it is very much time. Like, I think that, you know, there's so many people at UCL. UCL is so large, and so I think that part of it is actually just making time for people to, like, in their schedules, in their commitments and things like that, to have time to discuss these things. Because I think that one thing, you know, working between, I guess both Katharina and I work between two fields, and it takes a long time to learn two fields. Like, and so you can't just show up to say like, okay, well, I'm the kind of person who works in animals, and now I work in humans, and you're going to figure that all out. That takes years. And so I think that some of it is really having the capacity, and not like intellectual capacity, but just sort of like resources and time and support to do that. And I think that there just needs to be a bit more of that. Because it will take, I guess part of the issue is it takes a while for that to all play out to new and better findings and approaches and interventions and papers, which is, I think, what we all as a scientific community want. But that's something that is just, I think, a process, a slow process.
Yep.
And I think we might, now that we can meet in person as well, try and organize a few more sort of in-person workshops where we bring people together from different fields. Now that we've started to make these connections, I think it's always nicer to meet in person than online. So maybe we should organize that.
Great. Cool. Okay, well, I guess as we're kind of running out of time, I guess the last thing to do is just to note that the next seminar is going to be on the 10th of May, again from 1 to 12:15, and it'll feature talks by Jen Dixhuer and Jean Woltenscroft, and autism speaker Rina Anand, and addressing inequalities in children and young people's mental health. And so that promises to be really exciting. So please put that in your diaries. And then finally, just thanks again to both Tara and Katharina for 2 really exciting talks and really showing off like the breadth of excellence we have here at UCL. So thanks again, guys.
Thank you.
Thanks.